Okay, so as part of the microfluidics course, we have looked at the theory, and so we thought it would be beneficial to show you around the lab, the microfluidics lab that we have in the Department of Mechanical Engineering at IIT Madras. So we thought it would be beneficial to show what we do in the lab relating to the theory that we have studied in the microfluidics course. So here we do theoretical microfluidics. We look at the theory of different aspects of microfluidics. We also look at simulations, numerical simulations of different microfluidics phenomena, and we also do experiments. Okay. So what we will show you today is two things. One is to show you how we can make microfluidic devices. The first thing that we do is look at the theoretical aspects of certain phenomena that we want to investigate, and we also perform numerical simulations. So based on our preliminary studies and our simulation results, we come up with a CAD design that we use to fabricate the mask for the design of the microfluidic chip that we want to fabricate. And so we will show you the procedure that we adopt to fabricate microfluidic devices, and then we will also take you through how we perform experiments. Okay. As I was telling you, we first study the device structure that we want to fabricate, and then we come up with the mask layout, and we get the mask printed that you can see here. Okay, and this mask is used to follow the photolithography process to make a master, which we will be using to make the PDMS based microfluidic device. After we do the photolithography, so this is the master that we have fabricated using the photolithography process. So, it is basically SU 8 pattern onto the silicon wafer, which is SU 8 is a negative photoresist. So, it is patterned onto this silicon wafer using photolithography. So, this is the we are using the silicon master to make PDMS based microfluidic device. So, to make PDMS based microfluidic device, there are two components. One is the base, which is the silicon elastomer, which you see it here. This is the second component, the curing agent. So, we put this is the base which we discussed, and we use 10 parts of the base and mix it with one part of the curing agent. So, this is the 10 part of the base that we just poured onto the glass. We can use a plastic, disposable plastic glass. So, we have 20 grams of the base. We are mixing it with 2 grams of the curing agent. So, we have the base and the curing agent 10:1 ratio. So, then we mix the base and curing agent. So, we basically stir it. So, that is how it looks after the base and curing agent are mixed thoroughly. There are a lot of bubbles that you can see that are trapped. So, now we are going to do the degassing to remove these bubbles. So, this is the vacuum desiccator, which is connected to the vacuum pump, and we are going to do the degassing of the mixed base and the curing agent that we have seen earlier. After all the bubbles are removed from the mixture, this is how it is going to look like, is a transparent solution with no bubbles present. Okay, so this is the master that we have. So, it is a part of the master cut out. So, many times we have many different chips on a single wafer. So, this is part of the wafer which has one single chip, and we are going to pour the PDMS, that mixture that we have prepared onto this master. So, we have prepared an aluminum board, and inside that you can see the master, and now we are going to pour the PDMS onto this master. So, the thickness of the chip would depend on the quantity of the PDMS that we pour on the master. You can see some bubbles getting trapped during the pouring process, which will be removed during the baking that we will do in a vacuum oven. Okay, so after pouring the PDMS onto the silicon master, to remove the air bubbles and to cure the PDMS, we are putting the whole setup into the vacuum oven. So, the temperature is 65 degrees C, which is used for the curing of the PDMS. So, once the PDMS is cured for a duration of 3 hours at 65 degrees C, we take out from the oven one such PDMS setup which has been cured for last 3 hours. You can see it here. So, it is solidified. Now, we are going to peel the PDMS off from the silicon master. So, you can see that it has solidified, and now we are going to peel the PDMS from the silicon wafer. So, peeling has to be done from one end, and it has to be very gently pulled out. So, you can see that the pattern that was on the master has been printed onto the PDMS. Okay. So, the projection that was on the master has been formed as a channel on the PDMS. We are going to bond with glass to seal these channels, and to enable interconnection between the external fluidic supply, typically a syringe pump, and the channels in the microfluidic chip, we are punching the interconnection holes. So, you can see the punch which we are putting it through the PDMS layer to create the interconnection. Okay, so, these are the punch holes where the tubing will be glued to supply the fluid from the pump to the device. Okay, so after punching the holes, we are going to bond the chip with a glass slide. You can see that after punching the holes, the PDMS has been trimmed off to the shape of the chip, and it is going to be bonded with the glass slide, as you can see here. And for that, we are going to treat both the surface of the PDMS and the glass using oxygen plasma. So, this is the plasma system that we are using. It has basically 3 different parts. This is the main component. This is the plasma chamber, inside which we will have electrode, and the top one that you see there is a pressure gauge. It also has a flow meter, and on the left you would see a vacuum pump. It is a special vacuum pump. It should be noted that since we are dealing with oxygen plasma, we need a special vacuum pump to create the vacuum. Okay, because for ordinary vacuum pump, when we deal with oxygen, it may mix with the oil that we use in the vacuum pumps, which acts as explosive. So, there is a special oil free vacuum pump that is required in this case. So, we are going to bond the glass slide with the PDMS. So, they are put on a quartz surface and put inside the plasma cleaner. So, after the required pressure is reached, the plasma power is switched on, and the setting is at the medium level, and with slight adjustment of entrance of the oxygen into the chamber, you can create the plasma. So, we are going to treat the PDMS and the glass slide surface with the oxygen plasma for about a minute, and you can see the oxygen plasma. The coloration is a light bluish color that you would see. Okay. So, once the treatment is over, we take out of the plasma chamber and bond them. Okay. After the glass and the PDMS are treated, we take it out of the chamber, and put the PDMS channel surface facing down on the glass slide, and we apply a gentle pressure. So, with a very gentle pressure, the bond should be irreversible. Okay. So, the PDMS should bond with glass irreversibly. Okay. So, now the capillaries will be bonded to the chips on the punched hole areas, and so the capillary is inserted into the holes, and we glue it around. So, that is how the external fluid is going to supply it to the chip. Okay, so as I was saying, we are doing the theoretical analysis and experiments. So, this is one numerical simulations that we are performing. So, here there are two fluids of different viscosities coming into a channel, and we are trying to predict what the interface location is, knowing the viscosity ratio. Okay. So, this is a piezoelectric actuated valveless micro pump that we are trying to simulate. As you can see that in the forward stroke, as the membrane moves up, the fluid comes into the chamber more as compared to what it goes out, and in the reverse stroke, when the membrane goes down, more fluid goes towards the exit, out towards the outlet as compared to what goes out through the inlet. So, we have a net flow coming from the inlet to the outlet. So, this is a simulation of the motion of a droplet in a microchannel that we are trying to do here, and we are trying to predict the additional pressure drop that we encounter in the presence of a droplet as compared to in a case where we do not have droplet present in a channel. Okay, so here you can see we are doing an experiment on mixing two fluids in a microchannels. As you can see, we have two syringe pumps driving two different fluids at equal flow rates, and so the two fluids will mix, and there are going to be a diffusion zone which is going to be developed. So, here is the micro mixture chip that we are using for the experiment, and you can see there are two inlets and one outlet, and this the inlet fluidic connection is by the quarter 28 UNF thread, and we have these fluidic threads also connected to the capillaries, and which are threaded onto the fluidic port for the connection. So, this is the syringe pump that we use to pump the fluid into the microfluidic chip. We have a gas tight syringe, and this syringe pump is going to be driven by the stepper motor, and so this fluid is going to be pushed and going to go to the chip using this capillary. So, this shows the video that the experiment that we are running, where two fluids are being mixed, two colorless fluids are being mixed, and as they come in contact with each other, a diffusion zone is developed, and as you can see, the width of the diffusion zone increases along the flow direction. Okay. So, this is an experiment on generation of micro droplets in microchannels. As you can see, the continuous phase is trying to sear the discrete phase, and where discrete droplets are being formed in microchannels. We are also doing experiments related to particle setting. This is a mechanism called pinch flow fractionation, where we use a pinch segment to pinch a particulated sample with a pure fluid, and based on the principle that particles tend to move along the streamline passing through the center of mass, the bigger particles will be closer to the center line, and the smaller particles will move close to the wall, and that is how we will be able to sort particles using pinch flow fractionation.